Hello all, welcome back to Chinmoy's YouTube channel. So today we are here with a new lesson that is in plant physiology. You are having a germination of seeds. So let us start. Now what are the learning objectives of today's lesson? Now first we should know that what is seed. We should be able to classify seeds between the two types, that is monocotyledonous and dicotyledonous, exalbuminous and albuminous. Now we should know the structure and function of monocot as well as dicot seeds, taking suitable examples. So we are going to discuss with each and every example that what is the structure of a monocot seed and what is the structure of a dicot seed, with a label diagram. Obviously we'll discuss. Now diagram of a monocot seed and a dicot seed is very important because this diagram is for your ICSE exams also, for your final exams also, you are getting this diagrams of monocot and dicot seed. So let us start with today's episode of germination of seeds. Now first we should know that what is a seed. Now seed, as we all know, it is the first stage of the life cycle of a plant. So it is the first stage of a life cycle of a plant which contains an embryonic plant within and stored food material for the nourishment and a protective seed coat. So we can say that it is also a mature ovule after fertilization, and it is containing an embryonic plant, the stored food material for the nourishment and protective seed coat. Now seed contains a living plant in them that is called an embryo. So what is this? This is a seed contains a living plant in them that is called an embryo, and it contains a small amount of food to nourish the little plant. So very small amount of food is present in the seed to nourish the little plant. Now seed contains now seed coat. What is the seed coat? It is the covering. It protects the embryo from the mechanical damage and until it germinates. So seed coat is protecting the embryo from the mechanical damage and until it germinates full. Now what are seed leaves? You have already studied in your junior classes that seed leaves are called cotyledons, which are providing the necessary food until the roots and the true leaves are formed. So they are preserving the food and they are providing the necessary food until the roots and leaves are formed. Now what is embryo? The embryo inside the seed can remain at its dormant stage till it finds the favorable conditions to germinate. Now once it gets the favorable condition to germinate, it will grow out into the root and the shoot. So embryo is inside the seed and can remain in dormant stage until favorable conditions are coming to germinate. Conditions are present there to germinate. Now there are some seeds we can say that can remain dormant up to hundreds and hundreds of years in that seed state only, until and unless they are not getting any conditions to germinate. If they are getting conditions also, might be that is not the required conditions for the seed to germinate. So they remain in the dormant state up to hundreds and hundreds years or even more to germinate. So now let us see that what are the structure of dicot and monocot seeds and what are the types of different seeds. Now here we should learn about types of seeds. Now types of seeds, mainly we are discussing about 4 types of seeds, whereas first we will start with the monocotyledonous and the dicotyledonous. Now in your previous classes also you have already studied. So monocotyledonous and dicotyledonous we are discussing on the number of cotyledons present. So it is being discussed on the number of cotyledons present. So number of cotyledons present, monocotyledonous as the name suggests, that is 1 cotyledon, and dicotyledonous means that is 2. So dicot means it is having 2 seed leaves or 2 cotyledons. Okay, monocot means 1 cotyledon. Now mainly these seeds, they are not separated into 2 halves. Now this is the monocot seed. You will not be able to, if you soak the seed also for 12-24 hours also, you will not be able to separate the seed into 2 parts or 2 seed leaves. So these seeds, they do not separate into 2 halves like maize, rice, wheat, coconut and grasses. So once you soak it for like a long duration, 12 hours or 24 hours, when you press these seeds will be breaking down, but they'll not divide into 2 separate equal halves. Now the seeds like dicotyledonous seeds, these seeds contain 2 cotyledonous, So these dicot seeds, they contain two cotyledons, and seeds absorb and store nutrients from the endosperm before the seed germinates. So they are absorbing and storing the nutrients from the endosperm before the seed germinates. Now these cotyledons, they are usually thick. These cotyledons or seed leaves are there usually thick, and they store nutrients. So as they are storing, these cotyledons are storing the nutrients for the future use of the baby plant. They are thick in nature. Now what are the examples of dicot? Monocot, please remember these examples of monocot seeds because these are very common and they'll be coming in your exam. And the dicot seeds are pea, gram, bean, peanut and apple. So when you soak peanut and when you soak a pea, you can see when you press the seeds of a pea, you will see that they are divided into equal two halves. Gram also, you can see when you are pressing, when you are soaking that brown gram or brown chana, you are soaking that for 12 hours or 8 hours mostly. When you press that soft seed, the seed coat gets removed away and you will get two equal halves of cotyledons or seeds, which are the main representing that they are dicot seeds, or they are showing that they are having two seed leaves. So that is dicotyledonous seeds. So just go through the examples of this monocot and dicot seeds. Just remember that monocot seeds are unable to be separated into two halves, whereas the dicot seeds, they are equally divided into two equal halves, like examples of pea, gram, bean, peanut, apple seeds, etc. So let us see the next types of seeds. Here comes the division of the other two types of seeds, the albuminous and the exalbuminous. Now, how do we divide this exalbuminous and albuminous seeds? So that is being divided by the presence or absence of a special food storing tissue called the endosperm. Now, seeds are classified as albuminous and exalbuminous. Now, endospermic seeds, that is in which the endosperm is present and cotyledons are thin and membranous, that is the endosperm is storing the food. It is the special storing tissue for the seeds. So these seeds are called albuminous, in which these are endospermic seeds and in which endosperm is storing the food. Like for example, custard apple, poppy seeds are the albuminous dicot seeds, whereas the cereals, millets and palm seeds are albuminous monocot seeds. Okay, so these are albuminous dicot seeds and these are albuminous monocot seeds, that is they are having the endosperm which is being used for the special storing of the food. So here, as the endosperm is storing the food, the cotyledons are thin and membranous and they are not storing the food. Now here you can see the seeds, where the endosperm is there. This is the endosperm. You can see this labeling here. This is the endosperm which is storing the food for future use, but here the storage part is the cotyledon. So the cotyledon is occupying the thick and the fleshy part of the leaf, whereas here the cotyledon is very thin and very narrow, and endosperm is taking the whole region which is storing the food. So just refer to the diagram of the endospermic seed as well as the non-endospermic seed. So let us move on to the next part, that is the exalbuminous seeds. Now what are these exalbuminous seeds? These are the non-endospermic seeds, that is which lacks endosperm. Now cotyledons, there in the previous slide only I have shown you in the diagram, the cotyledons are very much thick and fleshy as they are storing the food, and there is no special endosperm present in the seeds for the storing of the food. So no special tissue is there. Now what are the examples? Gram, pea, mango, mustard seeds are exalbuminous dicot seeds, while Amorphophallus and Vallisneria seeds are exalbuminous monocot seeds. Okay, so gram, pea, mango and mustard seeds are exalbuminous dicot seeds, while orchids, Amorphophallus and Vallisneria seeds are exalbuminous monocot seeds. Now we should make a note of one term, that angiosperms, we all know that special thing, these are flowering plants, and gymnosperms, they do not have flowers, or they do not they neither do have flowers and nor they have ovaries. So this is just a note I mentioned while teaching you this exalbuminous and it is out of this lesson or out of this portions, just to know that angiosperms are flowering plants and are plants whose seeds are produced inside the flowers and are enclosed in protective structure called fruits. So flowers are there and the seeds are protected inside the structures called fruits. So fruits are protecting the seeds. Now gymnosperms, they do not have flowers or ovaries. So their seeds, how they are? Their seeds are like mature cones. Their seeds are like mature. They're maturing inside the cones. So these seeds, angiospermic seeds, they are maturing inside the fruits. They are being protected and covered by the fruits, whereas in the gymnosperms, the non-flowering plants, they do not have ovaries also. So their seeds mature inside the cones. Now here we are going to discuss about the structure of a seed. Now as we all know that there is a great variation in the size, shape and structure of the seeds. But the basic parts of the seed structure, they will be the same. So let us first discuss about the basic parts. So the first part, basic part is the cotyledon. So just now we have discussed about the cotyledons. What are these? These are the food storage organs that function as the first seed leaves. So what are these? These are the food storage organ. These are the food storage organs. Okay, so they are the seed leaves and they are acting as the first seed leaves. Now next comes the epicotyle. So what is the epicotyle? The first part of the embryo above the cotyledons is called the epicotyle, and that becomes the stem and the leaves of the plant. Now just follow where I am labeling in the diagram. So this is the epicotyle part. Okay, this is the epicotyle part. It is the epicotyle part in dicot leaf, and in the monocot it is the epicotyle part. So what it is doing actually, it becomes the develops into or becomes the stems and the leaves of the plant. So epicotyle, it develops into the stem and leaves of the plant. It is developing into the stem and leaves of the plant. Now in the dicot plant, this is the epicotyle, and in monocot, this one is the epicotyle. Is it clear? Just make it clear. Just I have given you the label diagram, and through this label diagram only it will be very clear for you all which one is the epicotyle. Now after that comes the plumule. So the epicotyle along with the embryonic leaves is called the plumule. So there are small embryonic leaves which are growing from the epicotyle, that is the plumule. Okay. So next comes the hypocotyle. So what is the hypocotyle? Here you can see the area hypocotyle. Hypocotyle. This one is the area of hypocotyle. So what is it? The area of the plant embryo between the cotyledons and the radicle. So between the cotyledon, this one is the cotyledon and this one is the radicle. So between the cotyledon and the radicle comes the hypocotyle part. So this part comes between the radicle and the hypocotyle. It comes between the radicle and the hypocotyle. This comes between the radicle, and it is between the radicle and the cotyledon. Sorry, it comes between the radicle and the cotyledon. Is the hypocotyle. So the part which is coming in between the cotyledon and the radicle in the seed, it is called the hypocotyle. Now plumule, I have already told what is plumule. That is epicotyle along with the embryonic leaves is called the this plumule. This epicotyle with this leaf is called the plumule. The epicotyle along with the small embryonic leaves is called the plumule. Now next comes the radicle. What is the radicle? We have seen here the radicle. The stem tip of the plant embryo developing into a root is called the radicle. So the part which is developing into a root, so it is developing into root. It is called the radicle. So the tip which is developing into the root is called the radicle. Now what is hilum here? Hilum, it is here you can see that there is a concave edge. There is an edge in both the seeds. We can see the edge is there. So that part where there is a white scar mark when it is attached to the ovary wall. So the white scar which is attached to the ovary wall. So white scar mark is called the hilum, which is attached to the ovary wall. Okay, scar mark attached to the ovary wall. It is called the hilum, attached to the wall of ovary. This one is called the hilum. Now what is micropyle? Micropyle, it is a very small opening on the concave side. Here there is a very small opening. This I'll give you in detail in the next slide, in the next few slides also. So there is a small opening that is in the concave side of the seed, which helps in the absorption of water as well as exchange of several gaseous things also takes place during germination. So this, when I am discussing with the structure of individual bean seed, I'll be discussing in detail. Just remember in your mind, when I'll be teaching the structure of a bean seed, structure of a maize seed, I'll be discussing this in full detail. So just remember, there is a small micropore that is called in the concave side of the seed, which is responsible for the absorption of water as well as exchange of gases during germination. That is called the micropyle. Now seed coat, as the name suggests, seed coat is the outside covering. So seed coat is the outside covering around the seed that protects the embryo and its food supply, that is the endosperm. Next comes the coleoptile. Now in the seeds, monocot seeds, actually this coleoptile is present. So what is coleoptile? It is actually a sheath that protects the plumule and it grows out of the soil. So that is the coleoptile, that is protects the plumule as it grows out of the soil. So these all I will be discussing when I am discussing the structure of a monocot seed, as well as everything in detail when I am discussing the parts of a dicot seed. Now next comes the endosperm. So on the basis of endosperm only we have discussed about the two types of seeds, that is the exalbuminous and albuminous. So we all know that endosperm is the food storing tissue. So we have already read in the previous slide that it is the food storing tissue which is helping the seed to store some food for future use, and in those seeds where the endosperm is present, the cotyledon will be thin. As cotyledon there is not storing food, there this special organ or endosperm is storing the food. Now is it clear to everyone? Now if you are having any doubts, please do ask in my comment box, and please do subscribe my channel so that you can keep on getting the updates. So let us move on to the next one. Now structure of a monocot seed, that is we have taken an example of a maize seed here. So see the structure of a maize seed. Now structure of a monocot seed, it can be studied by taking example of a maize seed. So maize seed actually it is a fruit, and in which the seed coat and the pericarp of the fruit wall are fused together, forming a protective layer. So here the seed coat and the fruit wall, what we are saying, they are actually fused together. So they are together. You can't differentiate the seed coat as well as the pericarp of the fruit wall. They are fused together, forming a protective layer. So that is why it is called a maize grain. So whenever the seed coat and the fruit wall are fused together, then only it is called a maize seed or a maize grain. So because the fruit wall and the pericarp, that is pericarp of the fruit wall and the seed coat, they are fused together. That is the seed coat and the fruit wall are fused together, which makes it a grain. So these two are fused together. Now if we are looking into this maize or grain or corn, what we will find, it is a small light colored oval shaped area at one side is there. Okay, so this area, it is marking the location of the embryo inside. So this oval shaped area, it is marking the location of the embryo inside the seed, and the remaining large part, the large remaining part, this part, what is it? It is the endosperm. So the large remaining part is the endosperm or the seed storing organ, whereas this part is the embryo, which is the light colored oval shaped area. So oval shaped area, it is the embryo, and the outside part which marks the location of the embryo inside the seed, that is the remaining large part, is called the endosperm, and it is actually it is lined with this remaining large part, it is lined by a large endosperm, that is called, this is it is an endosperm rich in protein called the aleuron layer. So what is it? It is the aleuron layer. Okay, so hence aleuron, what is it? It is the remaining large part in endosperm which is lined by a layer of endosperm rich in protein called the alluron layer. So this is the alluron layer. Now the endosperm, what is it rich in? The endosperm actually it is rich in starch, and we can say that a thin epithelial layer which is separating the endosperm from the embryonic part. So there is a thin layer which is separating the endosperm from the embryo. So thin layer is there which is separating the endosperm from the embryo. Okay, so the embryo in the maize grain, what is it is consisting of? A single cotyledon called the scutellum. So it is consisting of a single cotyledon called the scutellum. You can see the label diagram over here. Now these two diagrams are very important, so please do practice with labeling these two diagrams. Okay, this is the scutellum. Now where is the radicle and the plumule? So this one is the radicle and here comes the plumule. So the plumule is located in the upper side of the embryonic region, just opposite to the radicle. You can see they are just oppositely located. Radicle, it is actually situated down, and plumule is located. The radicle lies towards the pointed side of the embryonic region of the seed, and it is actually covered by coleorhiza. This is the coleorhiza which is covering the radicle, and the plumule is located just opposite to it in the opposite region of the radicle, and it is also enclosed by a thing that is called the coleoptile. So the radicle is covered by the coleorhiza, whereas the plumule is covered by the coleoptile. So these all new terms, this alluron layer, this scutellum, the embryo contains a single cotyledon called scutellum, a radicle, a plumule. So coleorhiza is protecting the radicle, whereas the coleoptile is protecting the plumule. So these all are very new terms to you all. So just learn these terms and try to label them in this diagram. So once you start labeling, you'll never forget that where is the coleoptile, where is the scutellum, where is the plumule, what is coleorhiza, what is radicle and all. So alluron layer. So you need to just learn this diagram, learn what I am teaching. Then only you can recall whenever you are sitting to study this. Now structure of monocot leaf, is it clear to all? Now we'll move on to the Now starting with the structure of a dicot seed, we have taken the example of bean seed here. So what is the speciality of bean seed? Actually it is like a seed which is easy to handle, and it is kidney shaped, and we can say that it is well demarcated by the concave and the convex sides. Now a bean seed is covered by usually testa. So this outer part, outer covering is the testa, whereas it is usually brown in color, and next to testa this part is the tegmen. So these testa and tegmen, they are protecting the inner parts of the seed. Okay, so these two layers, the testa and the tegmen. So the testa is the outer seed coat which is usually brown in color, and it is protecting the inside from drying out as well as from the mechanical damage. Now each seed is having some unique seed coats which is giving its appearance, distinctive appearances. Now lying next to testa is the tegmen. Okay, is it clear? Now it is a thin layer that also protects the inner part of the seeds. Now here you can see in this part there is hilum. So what is hilum? That is actually the area where the seed is attached to the wall of the ovary. And it is when you take the bean seed in your hand, you will see there is a white scar mark present on the concave side. So this scar mark is the hilum, whitish oval scar mark. So it is the area where the seed was attached to the ovary wall. So attached to the wall of the ovary. It was attached to the wall of the ovary. Now there below that only we have seen micropyle. What is the micropyle? The micropyle is the pore through which the pollen tube enters the ovule through fertilization. So if you have not gone through the video of fertilization, where I have already discussed the full process, how through the micropyle the pollen tube is entering the ovule and how the process of fertilization is taken place. So please refer to the I link above, as well as in my description box I am giving you the link of pollination as well as fertilization videos. Go through them. Now next comes the main two parts of the bean seed, that is a radicle and a plumule. So what is radicle? Radicle, it is forming that is root, while the plumule forms the stem leaves of the developing plant. So this is the This is outside is the seed coat, this is the radicle, this is the plumule part, and this is the radicle part. Okay, so these all are the main parts in the structure for dicot seed. Previous slide you have discussed about the monocot seed, and previous to that one we have in general we have discussed about the seed structure of a seed. Now here you can see that here this is the epicotyle part and this is the hypocotyle part. So previously we have told that what is epicotyle and what is hypocotyle. So epicotyle is the part which is above, just above the cotyledons, and hypocotyle is the area which is in between the cotyledons and the radicle. So that is the hypocotyle. So between the cotyledon and the radicle it is the hypocotyle. So it is between the cotyledons and the radicle is the hypocotyle, and the point of attachment of the cotyledon is also shown here. Okay, so this is how we are discussing the structure of a dicot as well as monocot seed. Please go through it. If you are having any queries, write in my comment box and refer to all the links which I am giving below, so that you can get a clear idea of plant physiology, basic biology and all the lessons which I have posted till now. So the next upcoming video is the seed germination in detail. So that is also there in your this lesson, that is which I have divided into a second part. So what are the conditions necessary for germination and the types of seed germination. So as of now I am concluding my video. Please go through it, subscribe, share as much as you can. Thank you.